Yes, the C3 pathway which we have discussed is taking place. That too, the first step, carboxylation, is taking place in presence of one very important enzyme. About that enzyme, they may ask the question, which is the enzyme which is abundant in nature. That enzyme is none other than rubisco. That enzyme is rubisco. That is ribulose biphosphate carboxylase oxygenase. Concerned to this, many times the question was asked, which of the following is the enzyme which is abundant in the nature? That is rubisco. That is ribulose biphosphate, carboxylase and oxygenase. Especially this enzyme takes part in the first step of C3 pathway. That is RUBP fixes CO2 to form two molecules of 3-PCA, phosphoglyceric acid. And now in some dry deciduous forest, the plants in dry deciduous forest, even the graminea members like maize and sorghum, jawara, we do find C4 pathway. C4 pathway instead of C3 pathway, where C4 pathway takes place. We have defined why the Kelvin cycle is said to be C3 pathway. That is said to be C3 pathway because the first formed stable compound is having 3 carbons. That is 3-phosphoglyceric acid. And C4 pathway means the first formed stable compound in this cycle is having 4 carbons. 4 carbons. That is none other than oxaloacetic acid. Oxaloacetic acid. Hence the name C4 pathway. The name is given C4 pathway as well. See here, in C4 pathway, the compound which fixes CO2 is phosphoenol pyruvic acid. It is having 3 carbons. It fixes CO2, forming oxaloacetic acid. That is OAA, having 4 carbons, and is said to be C4 pathway as well. With respect to C3 pathway and C4 pathway, 4 NEET question can be asked. In a C3 pathway, which compound fixes CO2? RUBP, having 5 carbons. And which is the first formed stable compound in C3 pathway? 3-PCA, 3-phosphoglyceric acid, having 3 carbons. In the similar way, name the compound which fixes CO2 in C4 pathway. PEP, having 3 carbons. Name the stable compound formed, first stable compound formed during C4 pathway, that is oxaloacetic acid, that is having 4 carbon. And I have just mentioned C4 pathway which is taking place in dry deciduous forest, and also the graminea members like maize, sorghum, even sugarcane. How this C4 pathway takes place? To study about that, we have to know about the anatomy of leaves of C4 plants. In C4 plants in the sense in which C4 pathway takes place. In C4 plants, the leaves do have a different anatomy. That anatomy is said to be cranch anatomy. That anatomy is said to be cranch anatomy. What is cranch anatomy? Already you know, in the leaves, the abundant green tissue which is named as mesophyll tissue, and the vascular bundle is present at the center, and that vascular bundle is covered by bundle sheath. In C4 plants, where the cranch anatomy is there, cranch anatomy is nothing but in the leaves, vascular bundle is covered by many layered bundle sheath with thick cell wall and possessing more number of chloroplasts. Is the definition. In C4 plants, the vascular bundle is covered by a bundle sheath, but it is many layered. But in C3 plants, where the vascular bundle is covered by a bundle sheath which is single layered, and here it is many layered with the thick cell wall and also large number of chloroplasts. That is cranch anatomy. In C4 plants there is a cranch anatomy, that's why they perform C4 pathway. So that it's time to study about C4 pathway. To study how the C4 pathway is taking place, we need to have the diagram. If you see the diagram, we can understand the process very easily. Even by seeing that sequence of reactions taking place, we can create many NEET questions. Yes. Let us see how C4 pathway takes place. For that, we need to have two cells. It's a mesophyll cell and is a bundle sheath cell, and both of them are interconnected by a protoplasmic connections. Protoplasmic connections between neighboring cells is said to be plasmodesmata. In mesophyll cell, when the CO2 enters in, that CO2 dissolves in H2O, forming carbonic acid. When CO2 enters into mesophyll cell, that is dissolved in water, forming carbonic acid. They may ask any general chemistry question. The weakest organic acid is, that is none other than carbonic acid. And what is carbonic acid? Even that carbonic acid is otherwise said to be aqueous CO2. Aqueous CO2. It means CO2 dissolved in water. That also can be asked in general chemistry question. Which of the following is a aqueous CO2? That is carbonic acid, and is the weakest organic acid. Is the weakest organic acid. And then this carbonic acid is unstable, dissociate to form proton and bicarbonate ion. Bicarbonate ion. In the mesophil, there is a 3-carbon compound, that is phosphoenol pyruvic acid. This phosphoenol pyruvic acid fixes bicarbonate ion in presence of phosphoenol pyruvic carboxylase. That is the enzyme which helps for the fixation of that bicarbonate ion by PEP. That enzyme is PEP carboxylase, leading to the formation, by the fixation, leading to the formation of C4 acid. Just mentioned that C4 acid is none other than OAA, that is oxaloacetic acid. It is having 4 carbon. This PEP having 3 carbon fixes bicarbonate ion, forming a 4 carbon acid, that is oxaloacetic acid, and is the first formed stable compound in a C4 pathway. And now the C4 acid through the plasmodesmata transported to the bundle sheet, where this C4 acid undergoes decarboxylation, removal of CO2. From this C4 acid, when CO2 is removed, the C4 acid becomes C3 acid. C3 acid. And C3 acid transported back to mesophyll cell. And this C3 acid is finally converted into phosphoenol pyruvic acid. The CO2 which is released here takes part in C3 cycle, that is Kelvin cycle takes place here in presence of Rubisco enzyme, leading to the formation of glucose. Why in C4 pathway that fixing of CO2 in the form of bicarbonate ion by PEP forming C4 acid, but it comes to bundle sheet cell where that C4 acid undergoes decarboxylation, removal of CO2, and becomes C3 acid. It goes back and becomes PEP, and the CO2 which is released enters into C3 pathway or Kelvin cycle, forming glucose. The question arises, the same C3 pathway could have taken place in mesophyll cell, but it does not, because Rubisco enzyme is absent in mesophyll cell. As Rubisco enzyme is absent, there is no chance for C3 pathway in mesophyll cell. That's why, by this mechanism, that C4 acids, they come to bundle sheet cell where decarboxylation and accumulation of CO2, and those CO2 molecules take part in C3 pathway where abundance of Rubisco is present. In this way, C4 pathway takes place. And here, many NEET questions can be created. The first NEET question is, in C4 pathway, phosphoenol pyruvic acid fixes CO2 in the form of bicarbonate ion. Answer is in the form of bicarbonate ion. In C4 pathway, the first stable compound formed is oxaloacetic acid. This C4 acid, when it goes to bundle sheet cell, is it there in the form of oxaloacetic acid? No, it is there when this oxaloacetic acid, it is further converted into that C4 acid, oxaloacetic acid is further converted into either malic acid or aspartic acid. Aspartic acid. But first formed stable compound is oxaloacetic acid, but is further converted into malic acid or aspartic acid and comes to that bundle sheath cell. This is about the C4 pathway, and one thing we should remember, Rubisco is the enzyme which is present in almost all mesophyll cells and bundle sheath cells in C3 plants, but in C4 plants that Rubisco is present only in bundle sheath cells but absent in mesophyll cells. And phosphoenol pyruvic acid, that phosphoenol pyruvic carboxylase enzyme, it is present only in the mesophyll cells of C4 plants but is absent in mesophyll cells of C3 plants. You remember that in C3 plants mesophyll cells that PEP carboxylase is absent. As it is absent, so that PEP never fix CO2 forming oxaloacetic acid. But in case of C3 plants, in whether they are mesophyll cells or bundle sheath cell, the enzyme present is Rubisco. And one more thing you should remember now only, the C4 plants, as far as the productivity of glucose is concerned, is very maximum than C3 plants, because here the accumulation of CO2 in that bundle sheath cells takes place very constantly. That's why the productivity is more in C4 plants and is less in C3 plants. Even C4 plants perform the process at high intensity of light. C3 plants perform the process at low intensity of light. Even C4 plants perform the process extremely at high temperature, but C3 plants perform at the low temperature. This is about C3 and C4 plants difference. And remember, many questions can be asked. Here, even coding and decoding also can be done. Mesophyll cell, this is, and is bundle sheath cell. Here A, B, C can be quoted. A is mesophyll cell, B is plasmodesmata, C is bundle sheath cell. Even here also they can make coding. PEP, A, HCO3, B, C, C4 acid. A is phosphoenol pyruvic acid. HCO3 minus, that is P, and C4 acid, that is C. Like that they can make the question. After this C4 pathway, let us discuss one more thing, that is photorespiration. Photorespiration. Before knowing photorespiration, let us know the first step in C3 pathway, where that RuBP having 5 carbon fixes CO2 in presence of Rubisco enzyme, forming 2 molecules of 3-phosphoglyceric acid. They are having 3 carbons. This is the first step. This rubisco enzyme, the name itself indicates, ribulose biphosphate, carboxylase and oxygenase. Its long form is ribulose biphosphate, carboxylase, oxygenase. What it means? That rubisco takes part in both carboxylation and also in oxygenation. It takes part in both carboxylation, fixing of CO2, even it also fixes O2. No doubt, rubisco enzyme is having more affection towards CO2 rather than oxygen. If CO2 concentration in the cell is more, this reaction takes place. In the stroma, this reaction takes place. If the O2 concentration is more, instead of this reaction, this RUBP fixes the CO2, fixes O2. If O2 concentration is more, that rubisco favors fixing of O2, leading to the formation of one molecule of 3-phosphoglyceric acid and one molecule of phosphoglycolate. This is having 3 carbon, this is having 2 carbon. Phosphoglycolate. Hence, instead of forming 2 molecules of 3-phosphoglyceric acid, just 1 molecule is formed. So that due to photorespiration, the process of Kelvin cycle is hindered, because 50% of the production of 3-phosphoglyceric acid is reduced if the O2 concentration is more. O2 concentration, if it is more, this rubisco enzyme, instead of helping for fixation of CO2, it helps for the fixing of O2. This is said to be photorespiration. They may ask the question, photorespiration takes place during which of the following condition? In the stroma, O2 concentration is more than CO2 concentration, it favors photorespiration. If CO2 concentration is more than O2, that favors the above reaction, so that Kelvin cycle C3 pathway takes place as it is. And remember, very important thing, photorespiration takes place only in C3 plants. But photorespiration does not take place C4 plants. It takes place in C3 plants, photorespiration. It does not take place in C4 plants, because in the C4 plants, in that bundle sheath cell, always CO2 concentration is more, because continuous accumulation of CO2 from mesophyll cell. So that no chance for the photorespiration in C4 plants. That's why their productivity is very high.